Here is the lecture for Chapter 8, the Cardiovascular System. The learning objectives for Chapter 8 are as follows. You need to be able to list and describe the layers of the heart walls. You need to know the chambers of the heart in detail and describe the path of blood flow through the heart. You need to be able to describe the structure and the locations of the various heart valves. You need to be able to differentiate between systole and diastole, and that includes both ventricular systole and diastole and atrial systole and diastole. You should be able to describe the process of cardiac depolarization and the repolarization of the cardiac cells. You should be able to describe the pathway of the electrical impulse generated by the SA node and how that impulse results in cardiac contraction. You need to be able to list and describe the unique anatomical and physiological features of fetal circulation. You need to be able to list the events responsible for the production of heart sounds heard on auscultation. You should be able to list the factors that influence heart rate and subsequently cardiac output. You need to be able to describe the relationship between cardiac output, heart rate, and stroke volume, and also know the 4 factors that contribute to the production of stroke volume. You should have a good understanding of the structure of arteries, capillaries, and veins. You should be able to list the major arteries and veins that travel between the heart and the various areas of systemic circulation. And you need to list the names and be able to describe the veins used for venipuncture in most animals. Your book begins with the composition of the heart walls. And when we talk about that, first thing we're going to need to talk about is the pericardium. Peri meaning around, cardium meaning heart. This is the layer that surrounds the heart. There is first of all an outer fibrous pericardium. The fibrous pericardium is made up of fibrous connective tissue. Even though it is a fairly thin wall, it is very strong. It protects the heart, and it loosely attaches the heart to the diaphragm. Inside, the next layer in from the fibrous pericardium is the serous pericardium. Serous meaning fluid, watery fluid. The serous pericardium is made up of 2 layers with a thin fluid-filled cavity between those 2 layers. The parietal layer directly attaches to, is adjacent to the fibrous pericardium. The visceral layer is deep to the fibrous pericardium and is also known as the epicardium. It lies directly against the heart itself. Epi meaning outside, cardium meaning heart. So we have a parietal and a visceral layer that are also making up the pericardium. Then from the epicardium inward we come to the myocardium, myo meaning muscle. The myocardium is located inside the sac formed by the pericardium. It is the thickest layer of the heart tissue. It is the actual heart muscle itself. This is the muscle that's going to do the work of pumping the blood out of the heart. Inside the myocardium is then the endocardium. The endocardium is a membranous lining between the myocardium and the chambers of the heart. The endocardium is actually a thin layer of simple squamous epithelium with a basement membrane. It is contiguous with all the great vessels that enter into the heart, so that as blood enters through the great vessels into the heart, there is always that smooth, nice, happy endocardium to keep those blood vessels, keep those red cells from becoming damaged, starting to form clots. The main purpose of the endocardium is to keep the red blood cells as they circulate through the heart very happy and intact. Here is another picture from a different book showing the layers of the heart. Starting on the outside, there is a fibrous pericardium. Then there is the parietal layer of the serous pericardium. And then there's the pericardial cavity itself. And that's where there is a scant amount of thin serous fluid. Then inside from the pericardial cavity is the visceral layer of the serous pericardium, which is also known as the epicardium. From there we can then see the endocardium, which is the thick heart wall itself. Then inside that is the endocardium, which is then going to be contiguous with the great vessels. This should show you that in one of the clinical applications for this chapter, when we start talking about cardiac tamponade, how that pericardial cavity could fill with fluid or blood and become a real detriment to cardiac function. We also need to mention here, and if you don't remember this, go back to the chapter on muscle tissue, that cardiac muscle is different than skeletal muscle and smooth muscle. It has some special features that the other cells do not have. Number one, cardiac muscle has a tendency to be kind of cube-shaped. Skeletal muscle are long, thin fibers. Cardiac muscle is much shorter and much thicker. The cardiac muscle is separated, cells are separated by intercalated discs, by cell walls. So this makes these cells one after the other, short kind of cube-shaped cells. Cardiac muscle also has a single centrally located nucleus, has some very good active gap junctions between the cells, and is a branching network instead of many fibers laid together like wires in a cable. This allows the heart to work as a functional syncytium. When one cardiac muscle cell depolarizes, then all the cardiac muscle cells will depolarize. So when the heart pumps, all the cells are working together. The intercalated discs, if you remember, also allow for the impulse to not only be spread evenly throughout the heart muscle, but it slows it, and therefore the heart contracts much more slowly. A regular skeletal muscle twitch is going to take about 1,000th of a second. A cardiac cycle is going to take about 0.8 seconds. So the cardiac muscle cells contract much more slowly. That allows them to work with force instead of just twitching and not pumping any blood. Again, here's a picture, cross-section of cardiac muscle. This is very, very similar to skeletal muscle, except for the length of the muscle. These are much shorter. The nucleus, instead of being peripherally located in skeletal muscle, is centrally located in cardiac muscle, but everything else pretty much works the same. We have sarcomeres, we have A bands, I bands, Z discs, Z lines, actin, myosin. We're going to have sarcoplastic reticulum. We're going to have calcium for this whole thing to work. We're going to have mitochondria to produce ATPs, so that if you don't have ATPs, the heart's not going to work. We have to have the energy to fuel this pump. One of the most important things that you should get out of this chapter, and you should remember from this course from this point on, is how blood flows through the heart. And so we're going to start a series of slides where we're going to talk about how blood gets through the heart. Number one, veins. Veins are vessels that carry blood to the heart, are going to collect deoxygenated blood from the tissues. This is going to be from the head, the trunk, the legs, and it's going to take this blood, which is fairly low in oxygen, high in carbon dioxide, towards the heart. The two vena cava, superior and inferior, are going to then carry blood into the right atrium. Superior from the head, the arms, parts of the chest, inferior from the legs and the abdomen. A third vessel actually takes blood to the right atrium as well. That's the coronary vein. The coronary vein is going to return blood from the heart itself to the right atrium. The opening for the coronary vein is the coronary sinus. So there are three openings into the right atrium: superior vena cava, inferior vena cava, and the coronary sinus. Once blood enters the right atrium, it's going to pass from the right atrium through the right atrioventricular valve. That is the proper name for that valve, the right AV valve. The slang or common name for it is tricuspid because it has three leaves. So therefore, the right AV valve, tricuspid valve are the same thing. However, the right AV valve is a proper name. After it passes through the right AV valve, blood then enters the right ventricle of the heart. Once blood has entered the right ventricle will constrict. A constriction is called systole, and then as it constricts, The pressure is going to close the right AV valve. That slams shut. Then blood must be forced out the pulmonary trunk through the pulmonary semilunar valves into the pulmonary artery. In the pulmonary artery, then the blood will split into either the right or left pulmonary arteries going to the lungs. From the right and left pulmonary arteries in the lungs, blood then passes through smaller and smaller and smaller vessels till it finally gets down to the capillaries in the lungs and the alveoli. We're then going to have the exchange of gases. Carbon dioxide is going to leave circulation, enter the alveoli. Oxygen from the alveoli is then going to enter into the blood, and we're going to get rid of the bad air, take in the good air, and that's where the exchange is going to take place. At that point, blood, which is then high in oxygen, low in carbon dioxide, we've gotten rid of the bad air, we've taken in the good air, is going to flow through pulmonary capillaries, start to come together into larger and larger vessels, and then finally we're going to end up with large vessels called pulmonary veins. Again, veins because it's going to the heart, and the pulmonary veins is going to bring blood rich in oxygen back to the left atrium of the heart. Once the blood is in the left atrium of the heart, it's then going to flow through the left atrioventricular valve, which is also, in human terms, most commonly known as the bicuspid valve. In veterinary nomenclature, it's more commonly known as the mitral valve. And from the left AV valve, it's then going to enter to the left ventricle. During ventricular systole, the mitral valve is going to slam shut. The left ventricle is going to constrict. That's going to close the mitral valve. Then it's going to force blood up the aorta through the aortic semilunar valve into the aorta, which is then going to go to the body. It's important to remember that each time the right and left atria constrict, they constrict together. Each time the right and left ventricle constricts, they constrict together. We don't have the right ventricle constricting and then later on the left ventricle constricting. Sometimes students get confused because as we describe blood flow through, they seem to think these are separate events. Atria constrict together, ventricles constrict together. Atria constrict together, ventricles constrict together. Then from there, blood's going to flow through the aorta. It's going to split off into smaller branches. Finally, it's going to get down to tissue capillaries. In tissues, oxygen is going to leave circulation, enter cells. Carbon dioxide from the cells are going to leave, go into circulation, and then we're going to start the process all over again where this blood low in oxygen, high in carbon dioxide is going to enter into the right atrium through the superior vena cava, the inferior vena cava, and the coronary sinus, and we complete the cycle all over again. This is a diagram from a different book showing essentially the same thing. If you look in this diagram on the right hand side down to kind of the lower middle area, they start with number 1, and this author starts with blood leaves the left ventricle of the heart, pumps out through the aorta and the aortic valve, goes to tissues, in systemic capillaries, blood loses oxygen, gains carbon dioxide. In this case, out goes the good air, in comes the bad air. Then that deoxygenated blood is going to return to the heart, number 4, through the superior vena cava, inferior vena cava, and the coronary sinus. From there, it's then going to go from the right atrium through the tricuspid or right AV valve to the right ventricle. It will be pumped out the right ventricle through the pulmonary valve into the pulmonary trunk, right and left pulmonary arteries. From there it goes to the blood, the capillaries surrounding the alveoli. Then good air is going to come in, bad air is going to go out. That's then going to collect into the pulmonary veins. It's then going to go to the left atrium. It's going to be pumped through the left AV valve or the bicuspid or mitral valve to the left ventricle. Same process is going to occur over and over and over. From there, your author spends quite a time talking about the external anatomy of the heart, going through the major structures. Number 1, there are auricles. Auricles are a part of the atrium. You can't really see atria when you look at a heart but you can see the auricles. They kind of have that elephant ear, kind of flappy area. They allow for stretching and overfilling of the atria. The ventricles, the left ventricle is a long, narrow. It's fairly thick-walled because it has to pump blood through the whole body. It terminates at the very apex or pointy part of the heart. The right ventricle is going to have a broader surface area. It will not be as thick-walled because it only has to pump blood to the lungs. And it kind of wraps around and surrounds parts of the left ventricle. The borders, or the separation, the septa, of the ventricles are separated by the interventricular sulci. Sulci singular, sulci plural, one on the left, one on the right. The sulci contain fat and blood vessels that are an integral part of the coronary circulation of the heart. That's where we're going to have a lot of the arteries and veins going into muscle tissue itself. Looking at the exterior of the heart, the cranial and caudal vena cava join together with the coronary sinus. They will then dump that blood into the right atrium. The pulmonary artery, if you're looking at an external view of the heart, emerges from the right ventricle as the pulmonary trunk, splits into right and left pulmonary arteries that go to each lung. The pulmonary trunk is larger and more curved than the vena cava. The aorta is pretty easy to figure out. It's the largest artery in the body. It's certainly the largest vessel, thickest vessel coming out of the heart. The walls of the aorta are the thickest of any blood vessel. It has the most pressure coming directly out of the left ventricle of any blood vessel. Has to be very thick, has to be very strong to handle that pressure. The aorta is going to emerge from the left ventricle into the aortic arch. The brachiocephalic trunk and the left subclavian artery branch off the aorta just after the aortic valve. Those are going to take blood towards the head and arms. Internal anatomy of the heart, we've talked about a lot about this as we describe blood flow, but we have, number one, an atrioventricular valve, a right AV valve, the tricuspid valve. It's called tricuspid because it has three flaps or three leaflets. The leaflets originate from a fibrous ring on the valve. Increased blood pressure in the atrium during contraction is going to force the flaps or the leaflets to open, and blood will be pushed out of the right atrium down into the right ventricle. The flaps are prevented from bending back into the atrium by the chordae tendineae, or strings that hold the leaflets in place. I have a good picture of the valves in a minute so you can see the chordae tendineae. The valves are one-way valves. They allow blood to flow from the right atrium into the right ventricle. They will not allow blood, under normal circumstances, to flow from the right ventricle back up into the right atrium. This picture doesn't show it real well. It's kind of hard to see. I've got a better picture coming up. But the chordae tendineae, little strings, connect the free edges of the flaps to the large, powerful papillary muscles. And then the papillary muscles attach to the interventricular septums. They attach to the wall of the myocardium itself. So we have three parts to any valve. We have the leaflets, we have the chordae tendineae, and we have the papillary muscles. Also, we have a moderator band. This is a specialized tissue that that is present in the right ventricle. It originates at the interventricular septum. It's not attached to the flaps of the tricuspid valve, but it does provide additional structural support to the wall of the right ventricle because the wall of the right ventricle is thinner. It doesn't have to pump blood very far, but it does need to be strong. So there is this moderator band, specialized tissue that does provide additional support to the right ventricle. Think of it kind of like putting rebar into concrete. Little extra structure there that makes sure things are going to hold in place. The left AV valve, mitral valve, also sometimes referred to the bicuspid valve, has two flaps, two leaflets. The outer edges of these flaps are attached to the fibrous ring. Inner edges are attached to the papillary muscles by chordae tendineae. Exactly the same setup on the left side as on the right on how the valves work. However, because the left ventricle is so big, so strong, there is no need for a moderator band. You don't find one in the left. The other two valves, they are the aortic valve and the pulmonary valves, also sometimes referred to as the semilunar valves. Each of these has 3 leaflets. They are attached once more at the outer edges to a fibrous ring around the vessel, and they are going to prevent blood flow out of the major vessels, the pulmon and the aorta, back into the heart, make sure that blood continues one-way traveling out of the heart itself. This is a picture again from another text showing internal structure of the heart. If you want a copy of this picture, let me know. I'll be happy to email it to you, or you can print it off from the PowerPoint presentation showing all the structures. I like this picture. It's very nice and colorful, shows everything you need to know, the major vessels, the major chambers, the structure of the leaflets, the myocardium, great vessels, really good picture of the internal structure of a heart. Here's the picture I promised about valves. This is a picture showing the bicuspid or mitral valve. Again, we have 2 leaflets. The leaflets are attached to a fibrous band in the atrium. They are then attached to the chordae tendineae, and then the chordae tendineae are attached to the papillary muscles. Constriction of the papillary muscles and relaxing is going to help open and close the valves. Also, pressures within the various chambers are going to open and close the valves to ensure one-way blood circulation through the heart. We need to cover the cardiac cycle, and to start with, we need some basic definitions. Systole is a term that means heart muscle is contracting. When heart muscle is contracting, blood is being ejected from one chamber into another. We can have atrial systole. When atrial systole occurs, blood is being forced from the atria down to the ventricles. When we have ventricular systole, we can then have blood being ejected from the ventricles up into the aorta and the pulmonary artery. Diastole is a period of relaxation when the heart is not beating. Again, we can have atrial diastole and ventricular diastole. Those two, either one of which, will be relaxed. We can have what is called cardiac systole, where the whole part of the heart is contracting, but we also have cardiac diastole when the heart is completely at rest. And in fact, half of a cardiac cycle is actually cardiac diastole. The heart is actively at rest. This diagram from another book shows a lot of the processes that are involved in a cardiac cycle. If you look at the very bottom down there, actually E, phases of the cardiac cycle, it shows, number one, we're going to have atrial contraction. When we have atrial contraction, blood is being forced out of the atria down to the ventricles. Then we're going to have isovolumetric contraction. Iso means same, volumetric means volume. The same volume of blood is going to have to be forced or pumped out of both the right the right ventricle and the left ventricle. So when the ventricles are contracting, they both have the same amount of blood in them, and they're both going to be forced out up through the aorta and the pulmonary trunk. Ventricular ejection is the actual forcing out. We're going to force the blood way up into those vessels with a lot of force. At that point, then we're going to have isovolumetric relaxation. Obviously, we're going to have both ventricles are going to relax. They are then going to fill. We're going to have atrial contraction, and we start the process all over again, one after the other. The second area up there, D, volume in the ventricles, shows how much blood is actually in the ventricles at any given point in this process. Obviously, just before they start to contract, they're going to be most full. That's when we're going to have the most volume. After they have actively constricted and the semilunar valves close, and just before the atria start to fill them up, that's when they're going to have the lowest volume of blood. Also, it's going to show heart sounds, how those are going to relate, and we'll talk about heart sounds more in a minute, how they relate to the various events that are occurring in the cardiac cycle. An important part of blood flow through the heart is the cardiac conduction system. The cardiac conduction system starts with the sinoatrial node, also known as the SA node, also known as the pacemaker. This is a specialized area of cardiac muscle cells that are located in the upper right atrium of the heart. These cells generate electrical impulses that will trigger the consistent and regular beating of the heart. Couple things you need to remember about the SA node. Number one, even though it is a bundle of true cardiac cells, it actually acts much more like nerve cells than it does muscle cells. It generates an impulse and conducts it. And number two, if left to its own devices, it will beat at a regular interval. It is like a governor on the heart. And we then have the sympathetic and parasympathetic systems that either speed up or slow down the pacemaker in response to various body needs. Part of the process of the cardiac cycle, part of the process of cardiac contraction, part of the process of the AV-SA node generating impulses is depolarization and repolarization. We've talked about this in cell membranes. We've talked about it in muscle cells. We'll talk about it more again when we get to nerve cells. But depolarization is the process of generating electrical impulses from the SA node. It results from the movement of cations, sodium, potassium, calcium, into SA node cells. A depolarization resulting, from the sodium potassium pump. This then causes a depolarization that then results in the cells constricting, that then results in systole, or active contraction of the atria. From there, we then have repolarization, or polarization, where once more sodiums and potassiums are pumped out of the cell or into the cell, results in the outside of the cell having a more positive charge than the inside of the cell. Remember, a resting membrane potential, we're going to have more sodiums outside, more potassiums inside, and then that is going to be a resting phase. That's going to be the diastole of the cell. Cardiac cycle occurs when an impulse is generated at an SA node. Once that SA node depolarizes, it's going to travel from one muscle cell to the next. It's going to have a wave pattern. It's going to cause all the atrial muscle cells to constrict. It's going to spread from the upper right quadrant of the right atrium. It's going to then spread down and to the left. So the right atrium actually constricts just a little bit before the left atrium will, because it's going to take a while for it to travel that far. All the atria constrict, then that's going to force blood through the right and left AV valves down into the ventricles. The impulse also is going to travel quickly down the muscle fibers, which act much like nerve fibers, to the atrioventricular node, also known as the AV node. The AV node is going to receive the impulse, and the impulse is then going to spread from the AV node down through the bundle of His. It's going to then travel down. It's going to split in right and left bundle branches. Right branch is going to go to the right atrium. Left branch is going to go to the left ventricle. Travels down the interventricular septum between the two ventricles to the very bottom of the ventricles at the apex of the heart. Once it reaches the apex of the heart, the fibers then travel up along the walls of the ventricle, and special fibers called Purkinje fibers carry the impulse deep into the muscle cells themselves. What's going to happen is when the SA or pacemaker fires, the atria are going to constrict right, then slightly left, and they're going to force blood down into the heart. Then, as the impulses travel down to the apex of the heart, once it turns and goes up the walls of the ventricle, the ventricular contraction is actually going to start at the apex. And so when it does, the impulse or the contraction is going to spread up the walls, and that ventricles are going to contract from the apex and force blood back up. Atria force blood down, ventricles force blood back up. And that specialized feature allows blood to be pumped from the ventricles down to from the atria down to the ventricles, and then from the ventricles up the great vessels to go to the lungs and to the body. In this cycle, the atria begin relaxation or diastole while the ventricles are still contracting. Pressure in the ventricles is higher than the pressure in the atria. That forces the AV valves to snap shut. The atria will then start to refill with blood passively. As the atria are becoming completely full, systole ends in the ventricles finish contracting. The aortic and pulmonary valves will then slam shut. The pressure in the ventricles fall below the pressure in the full atria. AV valves open, ventricles fill with blood, and then the SA node depolarizes again, causes the atria to contract and restart the cycle. Here's another picture from reference book showing the SA node, the fact that it spreads an impulse from the upper right atrium across the heart to the left atrium. Also sends impulses down to the AV node. From the AV node, it sends it down to the bundle of His. The bundle of His split into right and left bundle branches. They go to the apex of the heart. They turn. They then send fibers up along the ventricular walls. Purkinje fibers take that impulse into the walls of the ventricles. The ventricles will then constrict. It's important to know a lot of these structures because if you look at some of the references on EKGs and cardiac arrhythmias, we commonly have AV node problems. We commonly have right and left or both bundle branch blockage, and that's a very serious condition. Your book spends a good amount of time on fetal circulation. I think that's certainly important for you to know and to understand the basics of fetal circulation. Bypasses in the fetal circulation keep most of the blood out of pulmonary circulation. Certainly the fetus isn't going to be breathing. There's nothing there to breathe. So they're going to get their oxygen from the mother through the placenta, and that's going to require a very specialized circulatory pattern. In fetal circulation, oxygenated blood flows from the placenta through the umbilical vein. Blood from the umbilical vein then flows through the liver and the ductus venosus into the caudal vena cava. Then blood from the caudal vena cava is going to enter into the right atrium. Most of the blood from the right atrium is going to flow directly into the left atrium through a big hole between the two atria called the foramen ovale. Normally blood from the right atrium will flow down to the right ventricle, but because of pressure and because of the anatomical position of the foramen ovale, most of the blood from the right atrium is going to go directly into the left atrium through the foramen ovale. As we said in the last slide, most of the blood from the right atrium flows into the left atrium through the foramen ovale. Some blood will flow back through the tricuspid valve into the right ventricle, then to the pulmonary artery, but that is not really a significant or an important aspect of fetal circulation. Blood from the pulmonary artery flows into the blood to the lungs, or it will flow through the ductus arteriosus back down directly into the aorta. The ductus arteriosus is a tube that's then going to connect the pulmonary artery and the aorta. On birth, then that will close, hopefully. Blood flows through the fetal aorta to the fetal systemic circulation. That's just like in an adult. From the aorta, blood's going to flow down to body tissues that are going to need that good oxygen. At that point, we're going to repeat the cycle. Blood from tissue, oxygen from blood is going to enter into tissues. Carbon dioxide from tissues is going to enter back into the blood. And then deoxygenated blood is going to be sent back to the placenta through the umbilical arteries. At that point, it then re-enters mother circulation, and the usual stuff's going to happen. After birth, the ductus venosus constricts, and the foramen ovale and ductus arteriosus will all close. We should get closure on all those, and if we don't, then we do have big problems. Certainly we auscultate the heart, we listen, and so we do need to understand just a little bit about heart sounds. The old traditional cliche is that it makes a lub-dub sound, and it really does. First heart sound makes a good lub. The second heart sound makes a dub. The first heart sound, or the lub, is the closure of the mitral and tricuspid valves, the right and left AV valves, at the beginning of ventricular systole. They really do kind of slam together. They really do make a little click in there. The mitral valve is, under normal circumstances, the loudest on the left side of the chest. The tricuspid valve, or right AV valve, is logically best heard on the right side, since it is the right AV valve. The dub sound, or the second heart sound, is the closure of the aortic and pulmonary semilunar valves at the beginning of ventricular diastole. It's generally heard, those two valves are generally heard easiest on the left side of the chest. On the left side of the chest, in order, we generally listen to the pulmonic, the aortic, and the mitral valve. On the right side, we listen to the right AV valve. In large animals, and to a degree in humans, you can also hear two other heart sounds. The third heart sound is heard due to rapid ventricular filling. Blood, as it rushes down into the ventricles, does make a sound just like water going over a waterfall, and that can be heard in larger animals. And then also the fourth heart sound can be heard, contraction of the atria as they force blood down into the ventricles. So if you hear the third and fourth heart sound is rapid ventricular filling. The fourth heart sound is the actual contraction of the aorta, the atria, as they force blood down into the ventricles. Certainly one of the most important, and I think the most significant aspects of this chapter, is the concept of cardiac output. And I do want you to really understand cardiac output. It is absolutely essential to what you're going to be doing in many classes from here on out. Cardiac output is the amount of blood that leaves the heart. It usually is measured as the amount of blood that leaves the left ventricle in 1 minute. So by convention, cardiac output is the amount of blood that's pumped out of the left ventricle in 1 minute. It's going to be dependent upon primarily two things. There are two major things that make up the cardiac output. Number one is stroke volume. Stroke volume is the amount of blood that is ejected with each cardiac contraction. Every time the heart pumps, how much blood is actually ejected. That will go up and down. So obviously, if we have more blood ejected each time, we're going to have a higher cardiac output. If we have less, cardiac output is going to drop. Heart rate is also going to play a factor. Obviously, if the heart beats faster, then we're going to pump more blood. So we can vary stroke volume and heart rate to adjust cardiac output to ongoing body needs. A simple little diagram there, a little formula, is cardiac output is equal to stroke volume times heart rate. So if we know that the stroke volume was, say, 50 mLs and the heart rate was, say, 100 beats per minute, then our cardiac output would be 50 times 100, or 5,000 mLs per minute. Vigorous exercise results in increased contractility. In other words, the heart beats more forcefully. We end up with an increased stroke volume, or amount per beat, and we also often have an increased heart rate if we're going to start exercising rather strenuously. At this point, we always have to throw in Starling's law, because Starling's law says that increased filling of the heart results in increased force of cardiac contraction and increased stroke volume. In other words, if you're going to pour more blood into the heart is going to have to work harder to get it out, and it's going to have to get it out. What goes in must come out. So the heart will react to increased filling with increased cardiac output. Reduced blood pressure, such as in the case of shock. There's less pressure to fill the heart. We will have a decreased stroke volume. The heart rate frequently increases to compensate for the decreased stroke volume. So if the amount of blood going into the heart drops, the heart rate is going to increase to try to make up for that. The tissues out there are screaming for blood, oxygen, nutrients, and so the heart's going to try to work as hard as it can to compensate for that and to increase cardiac output. Cardiac output is going to be influenced by the autonomic nervous system. There are two parts to the autonomic nervous system. First of all, there is the sympathetic portion. The sympathetic portion is what we refer to often as the fight or flight portion. This part of the autonomic nervous system releases epinephrine or norepinephrine, and those two chemicals will increase stroke volume, they'll increase the force of contraction, and they will increase heart rate, make the heart beat faster. I do not like this analogy of general anesthesia. Many things will cause a stimulation of the parasympathetic nervous system. General anesthesia may or may not do this. So this is a very poor, this is from your book, and it is a very poor analogy. It's once more, I suspect, just between you and me, a textbook being written by people that don't really understand much about anesthesia. They get scared of it, so they blame it for every bad thing, up to including tooth decay and hemorrhoids. But anyway, the parasympathetic system, when it is stimulated, it is the opposite of the sympathetic system, it's going to have a tendency to slow things down. Its major neurotransmitter is going to be acetylcholine. And acetylcholine is generally going to result in a reduced stroke volume. As a general rule, heart rate may or may not decrease, but stroke volume will. This is a diagram from another book. This is a human textbook. You can tell that because there's a picture of a human being playing volleyball. This is probably one of the best diagrams you're going to see. At the top, we are going to have several concepts you need to understand. Stroke volume is going to be a factor of four things. They show three, there's really four. Number one, stroke volume is going to be a factor of preload, the amount of blood going into the heart. Number two is going to be contractility, how hard the heart actually constricts. Also, as a factor of contractility, you need to understand there's also a factor of relaxility. Relaxility means that for the heart to really work, not only does it have to contract hard, it has to fully relax. If it doesn't relax, the ventricles and atria cannot fill with blood. And then finally, there is afterload, and that's the amount of pressure that the heart has to work against to get blood out. If we have a patient with very high blood pressure, then the heart has to work much harder to get blood out against high blood pressure than, say, when there's low blood pressure. Increased end-diastolic volume, stretching of the heart, you'll end up with increased preload. We frankly do this a lot in veterinary medicine when we administer IV fluids. When we give IV fluids, we increase the preload, and we're going to increase stroke volume, and we're going to increase cardiac output, and we're going to make tissues happier. Positive ionotropic agents, such as increased sympathetic stimulation, epinephrine, norepinephrine, glucagon, thyroid hormones, calcium, and extracellular fluids, all those are going to increase contractility. Positive ionotropic agents, ionotropic means force of contraction, increase the force of contractions at all physiological levels of stretch. They're going to make it just work better. And then finally, decreased arterial blood pressure during diastole is going to result in decreased afterload. Therefore, less blood pressure, we're not going to have to worry about a lot of afterload. That's all going to go into increasing stroke volume. Those are all factors that we can influence with drugs and pharmaceuticals to increase cardiac function. Down at the bottom, we're going to have the nervous system, the cardiovascular centers in the medulla oblongata. We'll talk about that. It receives input from the cerebral cortex, the limbic system, proprioceptors, baroreceptors, and chemoreceptors. All that's going to increase sympathetic stimulation. It's going to decrease parasympathetic stimulation. We're going to get an increased heart rate. Heart's going to beat faster. Chemicals, the catecholamines, like epinephrine and norepinephrine, the same things are going to increase contractility are also going to increase the heart rate, and just some other factors. The very young, old, females, bad body fitness, couch potatoes, increased body temperature, you're getting hot. All those are going to often increase heart rate. If you increase stroke volume, if you increase heart rate, either or both of those, you will end up with increased cardiac output. Everything here, preload, contractility, afterload, relaxility, heart rate, all are things that we can control with pharmaceuticals and with drugs to moderate heart output, to increase it, to decrease it, to slow the heart, to speed the heart, to increase stroke volume, to decrease stroke volume, to increase preload, to decrease afterload. All that is stuff that we have control over. This diagram shows the cardiac center and the medulla oblongata of the brain. Remember that SA node, or the pacemaker, is going to be working at its own pace. It's like a governor. It's beating, it's beating, it's beating, it's beating. We can speed that up. We can slow that down through the cardioacceleratory center. The cardiovascular center is going to receive impulses from higher brain centers like the cerebral cortex, the limbic system, the hypothalamus. They're all going to be sending information to it. It's also going to be receiving information from sensory receptors, proprioreceptors, which is going to monitor body movements. Chemoreceptors are going to be extremely important. As hydrogen ion concentrations change, that's going to heavily influence the CV center. Also, baroreceptors, which monitor blood pressure, particularly in the carotid arteries, are going to send information to this center. On the basis of the information that is inputted into the cardiovascular center, it's then going to do one of two things. It's going to send information out to the cardioacceleratory nerves, the sympathetic system, which is going to increase force of contraction as well as heart rate, or it's going to try the brake and it's going to send out impulses through the vagus nerve. The vagus nerve is going to decrease the contractility to a degree, but mostly it's just going to decrease heart rate. And we can stimulate the vagus nerve to such a point that it slows the heart to it literally stops. This is a bad thing. Stopping the heart is a bad thing. No study of the heart is complete without studying a little bit about ECGs or EKGs. They mean the same thing. The original spelling was EKG, K for cardio. It was Germanic. It became anglicized, the C for cardio. They mean the same thing, just whether it's the more anglicized or the more Germanic old traditional form. Electrocardiograph is an instrument with a graph paper that moves under a stylus that produces a tracing. Electrical currents cause the stylus to move up or down as the paper rolls underneath it, so we can get an electrical snapshot of what is going on in that heart. And then there's three major parts to an EKG. Number one, as you see in the graph paper down there, there is a P wave. The P wave represents depolarization of the atria. The SA node fires and the atria depolarize. Shortly after the SA node fires, then the atria start to constrict. The QRS complex, that big set of waves, is the waves created by ventricular depolarization. Because the ventricles are so much larger than the atria, they're going to produce a much larger electrical event. The T wave represents the repolarization of the ventricles. There is no wave to show P wave or show the atrial repolarization that actually occurs during the QRS complex. You never see it. P wave is depolarization of the atria. QRS is depolarization of the ventricles. T wave represents repolarization of the ventricles. There is then a brief period of diastole, then we start the process all over again. Here's a graph from a different book showing much the same thing, a little bit more detail, showing P wave, QRS, ST, T, and in blue at the top it shows when the atria are actually constricting. That starts shortly after they start to depolarize. The yellow bar shows ventricular contraction. That starts very shortly after we start the QRS spike. That's when the ventricles are actually constricting. They do roughly correlate. Vascular anatomy and physiology, we do need to mention just a little bit about how the vessels themselves function. Blood in the systemic circulation is under higher pressure than blood in the pulmonary or coronary circulation. The systemic circulation is a high pressure system. You need more pressure to carry blood throughout the body. You need more pressure to carry blood all the way from the top of the head down to the tip of the toes. Blood in systemic circulation encounters more resistance to flow. You have more vessels, you have smaller vessels, you have, most importantly, more vessels in sheer length to get all that blood through. It has to go a long way from the heart down to the toes and then back again. The aorta is certainly the largest artery in the body. It is the largest in diameter and has the thickest walls. It has the most pressure. The artery wall layers are similar to the walls of the heart. They have a lot of connective tissue in there to allow for stretch. The aorta and pulmonary arteries, the middle layer contains more elastic fibers than muscle fibers, again primarily to allow for stretching. It allows the vessels to stretch slightly as they receive high blood pressure blood from the ventricles. They should stretch a little bit to accommodate for increases in volume, but they should not stretch so much to produce dilatation or low blood pressure. Your book talks some about vascular anatomy. Picture of the catheter, some good pictures showing systemic circulation. We won't worry a lot about that for lecture. That'll be something far more for lab. But the subclavian arteries branch off the aorta and travel towards the thoracic limbs. That's going to give primarily the blood flow to the arms. The carotid arteries branch off one or both subclavian arteries, depending on which species it is. They are going to send blood up towards the head and the brain. The main trunk of the aorta is going to arch, make a big curve, and then travels caudally, runs right down through the middle of the abdomen, through the diaphragm, along up against the spine, and then down to the legs. There are numerous branches from the thoracic and lumbar areas. We're going to have major branches: the celiac, the cranial mesenteric. Renals are probably going to be the most important because they do have to get to the kidneys. Phrenicoabdominals, circumflex iliacs. There's a whole bunch of branches that are going to come off that abdominal aorta until it gets down just about to the point where it ends. And the abdominal aorta is going to end in three major branches: the right and left iliac arteries that are then going to pass out of the body and become the femoral arteries to the legs, and the coccygeal artery emerges at the caudal aorta. It's going to continue on and go to the tail. If you've ever seen an adult animal that's had its tail severed, car doors used to get a lot, you'll find these guys put out a lot of blood. They put out a lot of blood fast, and that's because primarily it's just a straight shot off the aorta, high pressure system, and a coccygeal artery can put out a lot of blood in a short amount of time. Vascular anatomy, remember that smaller arteries continue to split into smaller and smaller and smaller vessels down into arterioles. Arterioles meaning small arteries. Blood flows through arterioles into tiny, thin-walled capillaries. Capillaries have no muscle layer in their walls. They're extremely thin. Therefore, it's easy for gases and nutrients to flow in and out of them into tissue cells, from tissue cells back into the capillaries. The capillaries then start to collect into larger vessels called venules. The venules will then collect into larger and larger and larger vessels called veins. Then the veins collect into the superior and inferior vena cava, and they return blood back to the right atrium. Venous blood is under lower pressure than arterial blood. Arterial blood is much, much higher pressure system. Therefore, the veins have thinner walls than arteries. They don't have to handle as much pressure. They don't have to be as thick or as strong. Because they are a low pressure system, veins do have valves to make sure that blood travels only in one direction of the heart. In most cases, just simple body movements, breathing, getting up, walking around, stretching your arms. All that is going to make sure that muscular contraction pumps the blood through the veins towards the heart. If it tries to flow backwards, the valves prevent it from going backwards, keeps blood going the way it's supposed to go. Veins in the foreleg will merge into larger and larger vessels to form the right and left brachiocephalic veins, veins carrying blood back to the heart. These carry blood to the cranial vena cava, back to the heart, right atrium. Veins in the hind limbs merge into the right and left iliac veins. These carry blood to the caudal vena cava. Caudal vena cava returns to the right atrium. So we have a big closed vascular system that's going to make sure the blood gets out of the heart to tissues and then back to the heart. There is smooth muscle in the wall of most blood vessels, certainly more in many arterioles than in any other. Constriction and relaxation allows the vascular system to direct blood to different regions of the body under different circumstances and in response to different needs. It is fairly easy to shuffle blood around from one area to the other, cut off circulation or limit circulation to one area, increase it to another. Depends on what's going on in the body, what the needs are, what the circumstances are. Certainly one thing we worry a lot about or concerned about or have to do is venipuncture. There's 3 species there: the cat, the dog, the cow. And certainly if you're working in a veterinary practice or have much experience with this, you're going to understand that all these are viable veins for blood collection. Certainly in the cat, we can collect from the cephalics, the jugulars, the medial saphenous. They call it the femoral with medial surface. It's really the medial saphenous. Those are all good ones. Probably in most cases, if you can get it from the jugular vein of the cat, that's your best option. The medial saphenous is number 2. Cephalics are a distant third. In dogs, jugulars, cephalics, and the lateral saphenous, and that's the worst drawing of a lateral saphenous I have ever seen. That is in exactly the wrong place, and somebody needs to go home and shoot somebody for drawing that. The jugulars are great places to draw blood, also great place for jugular catheters. The saphenous is a good place to draw blood, great place for lateral saphenous if you need a catheter there. We try to save the cephalics for cephalic vein or cephalic catheters. Those guys really work well. That's probably our optimal site for, in most cases, catheters in dogs. So if you want to get in trouble with me and if you want to get yelled at, draw the blood for pre-surgical or pre-anesthetic workup from the cephalic vein instead of one of the others too. And then when I go to put in a catheter, or you put in a catheter, or one of your workers puts in a catheter in the cephalic vein, and there's a big hematoma because you drew blood there when you didn't really have to because you had 2 other veins, then you're going to get in trouble. The cow, in the cow, there's 3 places to draw blood. Jugular, if you need large volumes, works great. The coccygeal vein, the tail vein right underneath the tail, is a good place for small amounts. Also, if you've got dairy cattle, Holsteins, Jerseys, you can use milk veins, especially if they're lactating, along the lateral side of the lower abdomen. That's a good site if you're working down there. Horses, in horses we draw blood from jugulars, and preferably from the right jugular. In sheep, goats, we also are looking primarily at jugular veins. Those are the good places. Also in cattle, some people can, if you need a very small amount of blood and you're working to the head, you can get one from the ear vein or the auricular vein. Those are all options for getting blood. And this is once more talking about the same thing. Jugular veins, we talked about those. Especially in large animals, the jugular vein seems to be right up against the carotid artery. Got to be careful when you're dealing with there. Every so often, particularly in horses, we'll go and we'll get a little enthusiastic, especially students, get a little enthusiastic about getting through that horse skin, and they'll go right through the jugular vein into the carotid artery, and boy, that thing will pump out a lot of blood in a big hurry. Really gets people's attention. And again, talking primarily about the caudal epigastric vein, milk veins, particularly in dairy cows, coccygeal vein. We use it a lot to bleed for, like Bangs testing and some other stuff. Your book does a good job of talking about clinical applications, and there's some really good clinical applications. Hardware disease, fairly common in cattle, dairy cattle. These guys will go out and as they graze, they'll pick up small bits of metal, nails, little pieces of wire. Those fall forward into the reticulum of the ruminal system of the stomach. They can then puncture through the diaphragm and cause a traumatic pericarditis. I kind of don't like this clinical application in this part because to me this is more of a digestive disease than a cardiovascular disease, but hardware's here. Pericardial effusions, cardiac tamponade, we don't see that much in veterinary medicine. It does happen. Very characteristic set of physical signs, very characteristic, and a wonderful radiograph on these guys. Put them on a table, take an x-ray, and it looks like they have a bowling ball or a basketball in their chest. This big, huge, round thing as the pericardial sac fills with blood or with fluid. It's much more common in fairly common in humans that decide to hit things going fast in cars and not wearing seatbelts and smashing their chests into steering wheels. Patent ductus arteriosus, you can read through the information on PDAs. PDAs, one thing that's interesting for me is that with PDAs, you're going to find that every so often people will show up with a puppy, particularly. The magic range is 8 to 12 weeks, about the time of showing up for shots. They will say, little puppy is not doing good, doesn't have a lot of energy, poops out a lot. You look at them, they're very blue, do not have good oxygenation, do not have perfusion, just the traditional failure to thrive puppy, just don't do well. And then if you stick a stethoscope up to their chest, all you hear is they have a very characteristic murmur called a machinery murmur. It's a holosystolic murmur. It just goes on and on and on and on. One of the suggested websites for the week is one that has heart sounds in it, and you can hear a PDA if you go to that site and listen. Once you hear one, you'll never forget them. Defibrillation. We don't do a lot of defibrillation in veterinary medicine, particularly because we just don't. We just don't have defibrillators. We don't monitor very well. We just don't do very well. Finally, congestive heart failure. Congestive heart failure is one of the most common things we do see. Very common in dogs. These older dogs show up, have congestive heart failure. There's a whole host of things, pharmaceuticals that we can talk about due to that'll help them. Diuretics, positive chronotropic, inotropic drugs. One of the things that has really changed in veterinary medicine over the last few years is our ability to treat congestive heart failures. These dogs used to have, even as far as 15 years ago, congestive heart failure was just a death sentence. They were going to suffer. They were not going to do well. All we could do was digitalize them. That was a big mess, not a pleasant experience. And now with the newer modern generations of drugs like enalapril and some of the others, we can really do a good job of managing these guys. We can buy them a lot of time, sometimes a year or two, high quality of life. We're not going to fix it, but we are going to make it a lot better to the point where they can enjoy life once more. Congestive heart failure is really one of the most fascinating things we talk about in veterinary medicine. I suggest you read that, and if you want more information, there are some clinical case studies in your suggested PowerPoint and your suggested websites for this week. They can give you in-depth and detailed information about how to manage and treat congestive heart failure.